Hello students, let us discuss the different modes of asexual reproduction. First, fission. Fission means division of the cell, and fission usually occurs in unicellular organisms and that too in protozoans. Fission can be either binary fission or multiple fission. Binary fission means division of the parent cell into two. Binary fission can happen in two ways. Either by a single plane passing through the center, as in the case of Paramecium, Euglena or Leishmania, or it can happen through any plane passing through the center, as in the case of amoeba. Then what is the difference between Paramecium, Euglena or Leishmania? In Paramecium, the fission is transverse, while in Euglena and Leishmania it is longitudinal. Now let us discuss activity 8.3. We need to observe a permanent slide of amoeba showing binary fission. When we observe this, we see that the nucleus elongates and it divides. This is known as karyokinosis. This division of the nucleus is followed by division of the cell. This division of the cell is known as cytokinesis, and the division of the nucleus takes place mitotically. After the division of the nucleus and the cell, two identical daughter cells are formed. So we cannot differentiate between the parent cell and the daughter cell. Then what is multiple fission? Multiple fission means division of the parent cell into many identical daughter cells. Example for this is Plasmodium, which is also a protozoan and malarial parasite. Next, let us move on to fragmentation. Fragmentation is seen in Spirogyra, which is commonly called pond silk. Let us refer to activity 8.4. We need to collect some water from a pond or a lake which is dark green in color and has filamentous algae. We need to take one or two filaments and place it on a glass slide, put a few drops of iodine and place a cover slip on it and view it. When we view it under the microscope, we can see the detailed structure of the cell of Spirogyra. There are two cell walls. The outer cell wall is made of pectin and inner one is made of cellulose. The outer cell wall dissolves in water and becomes mucilaginous. Inside the cell wall is present the cytoplasm, and suspended in the cytoplasm are the nucleus, the ribbon-shaped chloroplast, vacuoles, mitochondria, Golgi apparatus, etc. The nucleus is kept in its place by cytoplasmic strands, and the characteristic or the identifying feature of Spirogyra is the ribbon-shaped chloroplast on which are present starch granules called pyrenoids. Now Spirogyra is a filamentous algae. These filaments are long and thin, made up of many cells. Each cell is longer than broad. When the filament matures, it breaks up into small pieces, and each piece is known as a fragment. These fragments then develop into new filaments. This is known as fragmentation. Now, why is fragmentation not possible in higher order organisms, because higher order organisms are not a random collection of cells. Instead, they have a well organized body with specialized cells, tissues, organs and organ systems. Now let us move on to regeneration. Regeneration can be seen in organisms such as planaria, hydra, starfish, etc. Sometimes accidentally these organisms get cut up and each piece develops into a new organism. Then students, what's the difference between fragmentation and regeneration? In fragmentation there are no specialized cells, but in regeneration there are specialized cells called regenerative cells. These regenerative cells undergo rapid proliferation and form masses of cells which differentiate into special tissues, and thus there occurs a sequence or an organized sequence of changes which result in development. Then why regeneration is not reproduction? Because an organism cannot depend on being cut up to reproduce. Now let us move on to budding. The two examples we have to study here are hydra and yeast. First let us take hydra. Hydra, it develops a small protuberance. The protuberance develops by the continuous division of the regenerative cells, and these protuberances are formed at specific spots which undergo division continuously, and these protuberances mature into buds. These buds, when fully mature, they detach from the parent body and develop into new individuals. Now let us refer to activity 8.1, where we need to make a culture of yeast, that is by dissolving a little bit of sugar in water, and to this we need to add a pinch of salt and keep it for 2 to 3 hours. After this, we take 1 or 2 drops of the yeast culture solution and place it on a glass slide. Pour a drop of glycerin and place cover slip on it and view it. We can see the different stages of budding in yeast. Now yeast belongs to fungi, and yeast is a unicellular fungi. That means it has only a single cell. The yeast cell develops a small protuberance. This protuberance then develops into the bud. The nucleus then divides and migrates into the bud. Eventually, the bud matures and detaches from the parent cell. This division is asymmetric. So, here there is a stark difference between the parent cell and the daughter cell. We can differentiate, or in other words, we can differentiate between the parent cell and the daughter cell. Thus, budding occurs in yeast. Now, what is the difference between budding in yeast and hydra? In hydra, there are specialized cells called regenerative cells, which help in the formation of the bud. But in yeast, there are no such specialized cells. Secondly, in yeast, a chain of buds are produced. But in hydra, only a single bud is produced. Now, let us move on to vegetative propagation. What is vegetative propagation? New plants are formed from any other part other than the seed. That is, it can be formed from stem, leaves, roots, etc. Now, vegetative propagation can be done naturally or artificially. Examples for natural vegetative propagation is through stem, leaves, bulbils, bulbs, corm, rhizome, stem tuber, etc. While artificially, it can be done by grafting, layering and tissue culture. Now, what are the advantages of vegetative propagation? We have to discuss three advantages. The first one is that the plants that are formed from vegetative propagation, they bear fruits and flowers at a faster rate than the plants which are formed from seeds. Secondly, some plants have lost their capacity to produce seeds. Such plants can utilize this method. For example, in the case of banana, rose, jasmine, etc. And the third advantage is that the offsprings are genetically similar to the parents. The disadvantage is that new varieties cannot be formed. Now let us see how vegetative propagation occurs in potato, which is an underground stem and is known as stem tuber. Let us refer to activity 8.5. If you observe a potato, you can see notches. Each notch contains two nodes and between the two nodes are present a bud. We have to cut this potato into small pieces such that some of the pieces have the notches and some do not. We keep these pieces on a cotton in a petri dish and keep it moistened and keep this experimental setup for 2 to 3 weeks. We can observe that only those pieces which have the notches develop small leaves and roots. The other does not. This proves that the buds help in vegetative propagation. Next, let us see how vegetative propagation occurs in bryophyllum. The bryophyllum leaf margin has notches. In the notches develop buds. These buds, when they mature, they develop leaves and roots. When these small plantlets, they are mature enough, they fall off to the ground and develop into new plants. Now let us see how vegetative propagation occurs in money plant. We have to refer to activity 8.6. We need to take a piece of money plant and cut it into smaller pieces such that some pieces do not have any leaves while some pieces have at least one leaf. We need to keep these pieces in a bowl containing water for 2 to 3 weeks. After 2 to 3 weeks, we can observe that only those pieces which have leaves develop roots and small leaves or show the tendency for development. The pieces which do not have any leaves do not show any changes. This is because when the leaves are present, photosynthesis occurs and this provides energy for further development. And those pieces which do not have the leaves do not get any energy, so they remain as such. Thus we can see how in money plant vegetative propagation occurs through stems. Next, let us discuss tissue culture. Tissue culture is a modern method of vegetative propagation. Here, from the shoot tips, tissues or cells are extracted and it is grown in a medium where these cells develop rapidly or proliferate rapidly and develop a mass of cells called callus. This callus is then transferred to a solution of hormones where they undergo growth and differentiation and develop into small plantlets. These plantlets are then transferred to the soil where they grow and mature. The advantage of this method is that many plants can be grown at a time in disease-free condition. This method is especially used in the case of ornamental plants. Next, let us see what is spore formation. Spore formation is usually seen in fungi, bryophytes, pteridophytes, and even in algae. Let us refer to activity 8.2. We need to take a piece of bread and moisten it and keep it for a week. After a week, when we observe it, we can see black velvety structures. When we take a magnifying glass and observe it properly, we can see a few things. Let us discuss that. Thread-like structures which are found on the bread or which are formed on the bread are known as hyphae. And from the hyphae develop stick-like structures called sporangiophores. On the tip of each sporangiophore is tiny blobs. These tiny blobs are known as sporangia. The sporangia contain spores. When the sporangia matures, the sporangia burst and spores are released into the air. Spores are specially adapted because they are very light in weight, which allow the easy dispersal through the air. Not only that, they are well protected by hard walls or hard outer covering, which help them to overcome unfavorable climatic conditions. When they fall on suitable substratum, they regenerate. Thus, we have discussed all the modes of asexual reproduction. Let us once again bring to our memory what is asexual reproduction. It is a method in which only a single parent is involved. There is no gamete formation and the offsprings are exactly identical to the parents. Thank you.